Hey, thanks very much for the invitation to speak here. So I come much more from the microbiome side of things. I'm assistant professor at the Jackson Laboratory, and my slides aren't showing. I'm sorry? Ah, okay, there we go. All right, yes, so that's me. That's Jax. You heard from Ed earlier this morning. So in my group at the Jackson Laboratory, we use primarily shotgun metagenomic sequencing to reconstruct these microbial communities in terms of who's there, so the composition, as well as the function. So what are they doing? And so basically what we do is we take microbiome samples. So here a skin swab. I do a lot of my work in the skin microbiome. We make a bulk sequencing sample, then sequence it very deeply, and then use different computational approaches to reconstruct draft metagenomes that in essence represent the different species, the original species in the skin microbiome, and in essence creating a kind of a high resolution parts list of the genes and the genomes in the skin microbiome. And so with this kind of data, then what we can do is we can create these very high-resolution compositional maps of the skin microbiome at many different skin sites. So these are the different sites shown here. And what the consensus pie charts are showing are basically the different viruses, the fungi, the bacterial species at that particular skin site. And so one of the striking findings about the healthy skin microbiome is this very strong site specificity, where the composition and as well as the function of the microbial community is really very strongly driven by the physiologic characteristics of the skin. So for example, whether it's an oily site like our face or our torso, moist like our feet, or dry like our hands and forearms. And so you can see just by eye the heterogeneity in the composition of the pie charts in the figure there. When there's skin disease, what we see is a very significant shift in kind of these core characteristics of the site specificity. So this is just a snapshot of a study I did earlier looking at the microbiome of atopic dermatitis or eczema, so one of these itchy inflammatory skin conditions. What we saw was that during a disease flare-up, these atopic dermatitis patients lost their natural biodiversity that's much more typical of the healthy skin, which you can see here in this control plot here, and with a very significant increase in the relative abundance of the staphylococci here in blue. And so from our studies of the skin microbiome and from those of others in the field, we converged on a central etiology for atopic dermatitis, where the skin disease could be initiated, exacerbated, or recur by three major mechanisms driven either by microbial pathogens or dysbiosis, or these imbalances in the skin microbiome, defects in the barrier defenses of the skin, or actually a hyperallergenic state where the immune system is responding too strongly to the different stimuli. And so this was kind of our test bed for developing a new type of therapeutic for AD, which is typically treated with things like antibiotics, steroids, things that don't work very well. There's a very high recurrence rate for this disease. And so what we set out to do was to create an engineered probiotic to address these major disease factors. But before getting there, we came on some unique challenges. And among these was what I started out with, which is the skin site specificity. So our ideal microbial chassis has to be able to compete against many different existing communities, and at the same time also be stable, as well as have effective penetrance and stability in the skin. So in the end, we had to come up with a new microbe, but identify a microbe that would make a good chassis. And so our first thought was to use one of the microbes that's already common in the skin, because we know that it can colonize the skin. Moreover, it's generally safe if it's human-derived. And so our first criteria was to identify a skin microbe that could grow in most of the different skin sites. And so the way we did this is because we don't actually have a lot of hands-on knowledge about the different microbes in the microbial communities of the skin, mostly because it's very difficult to culture or manipulate human-associated microbes. And so what we did is to take our metagenomic data and approximate microbial growth rate. And this is just based on the fact that bacteria that are growing very rapidly will have greater coverage of the DNA around the replication of origin where the bacteria are initiating DNA synthesis. And so then by looking at coverage of metagenomic reads across a genome, you can detect this by looking at the ratio of the highest coverage here to the lowest coverage there, where the high peak to trough ratio therefore indicates a much more rapidly growing population, in contrast to a PTR of basically 1 or flat right here, basically meaning that the microbe is not dividing or could actually be dead. And so we did this for a lot of different microbes across the different skin sites. And what we found actually, surprisingly, is that in a large proportion of skin sites, most of the microbes or many of the microbes were actually stationary or dead, and that relatively few microbes were active in most skin sites. And among which was our microbe, this microbe here on the right or left, Staph epidermidis. So maybe this would make a good candidate. It's active in most skin sites. It grows relatively readily. But the second challenge of using one's own microbes is that it also has to successfully compete against a lot of its own kind in the microbial community. And so what I mean by that is that for Staph epidermidis, not just the Staph epidermidis species exists in the skin, but multiple different strains of Staph epidermidis actually also exist. So what we've done is to develop different computational tools to also reconstruct strain diversity in addition to species diversity. And this is just a relative abundance plot of the different Staph epidermidis strains here. So each color here represents the relative abundance from this Staph epidermidis phylogenetic tree. And then we're looking at a time series here. So time point 1 to 2 is about a year. Time point 2 to 3 is about a month. And so each of these sets show, first of all, is that we have a lot of different Staph epidermidis strains in different skin sites, but also that they're pretty stable over time. So we're actually, my given Staph epidermidis community is actually relatively refractory to obtaining new Staph epidermidis strains, for example, from the neighbor that I was sitting next to earlier. So what we've done is we've gone back to the basics, and we really want to identify features of Staph epidermidis, different strains that would make it basically a very effective colonizer in different environments. And so what we're doing is we're using different computational approaches to understand how these different strains are different, identifying genetic features that we can then engineer with CRISPRs and synthesis and approaches to really, in essence, engineer a Staph epidermidis that has genetic characteristics that makes it a more universal but also competitive colonizer. Because the implication here is that the genetic diversity that's encoded by all of those different Staph epidermidis strains, each has given it some kind of benefit in terms of survival in the skin community. So we're really trying to harness those beneficial characteristics in order to make a super Staph epi strain. And so then for a disease like AD, we want to address our AD triad of the microbe, the skin defects, as well as the immune system. And so what we're doing is we're engineering our new Staph epidermidis to have a modular system so that we can then swap in and out different therapeutic parts, which in this case might be, for example, an anti-Staph aureus biosensor, secrete different therapeutic proteins, or different anti-inflammatory molecules. And this is my final slide here. More generally, I just wanted to throw out there that my goal here is really to combine both metagenomics as well as synthetic biology, where the goal is really to arrive at a point by we're able to characterize the microbial community, input many different characteristics of a microbial community using our shotgun sequencing, our computational approaches, to really then be able to create a mathematical model by which we can use this information about the characterization of the skin or the microbiota that's there to really both identify and optimize potential engineered probiotics, but then also predict the efficacy of its integration, as well as predict its impact on the host as well as the microbial community. So that's it. This is my lab at the Jackson Laboratory, and thanks very much also to my collaborators there. Okay.
Very interesting. I was wondering if one of your goals is to go to the clinics. What are the odds that aureus will become a hypercolonizer, or that epidermidis will become a high pathogen?
So they kind of swap roles. That's a great question.
Transfer.
And many people actually who have spoken before me today have addressed that in terms of making them less compatible. For example, some of the codon engineering, I think, would be a very effective way to do that. I mean, so right now we've done, I think, the easier things like make kill switches to, target our recombinant microbe. But, again, that doesn't prevent things like horizontal gene transfer. And we're actually trying to just biologically characterize what is the maximum rate of HGT between different staphylococcal species, just to understand what would be the maximum possible risk. Yeah.
So you're optimizing this chassis as like a biosensor and a delivery vehicle. How do you test it? And you can't grow it. Can't grow it. What do you find patients to do experiments on, or what are you gonna
So
do?
the nice part about staph epidermidis, unlike some of the other skin microbes that I mentioned, is that we can actually cultivate and grow it. So we're actually doing in vitro as well as mouse studies to check out the activity of the things that we're putting in. So okay, all right, thanks very much.